This is Dr. Ed Park. I'm recording Lecture 7 out of 9 in this series on exosomes, and this one is entitled, Are Exosomes FDA Approved? Well, of course, the answer is no. And let's talk about really a more interesting question, which is, what does it mean to be FDA approved? Now, if you really think about it, the FDA's mission represents a lot of really good things. They're trying to prove efficacy, they're trying to prove safety, they're trying to maintain quality control in the manufacture of certain drugs. And this is all very good and well in the service of consumers and patients. Now, the FDA is a regulatory body. It is also an intellectual property oversight agency. And in that aspect, it works to protect corporate interests. In other words, if you pay the money to go through an FDA trial, then it's like the teachers being paid by the students. The whole thing is funded by the drug companies. So there is an inherent bias towards newness. There's no point in reinventing something that is already off patent and available freely, either generically or even over the counter. So there is this drive towards newness, intellectual property, and again, maybe we don't want a better Frank Sinatra. We just want the original, and maybe in terms of like the return on investment, a lot of these newer drugs are not going to be better. They're just going to be better for certain shareholders. Remember that drugs are usually just blockers of certain pathways. So in that sense, they're disease mitigators. Oftentimes they create an imbalance of something that is naturally balanced. And so they do nothing to really prevent disease in most cases, with antibiotics being, of course, the exception. Antibiotics conspire with your immune system to overcome a temporary problem. So much of the diseases that we have are actually chronic cellular dysfunction, I would maintain, based on stem cell mutation and epigenetic changes. So if you want to understand more of that, please watch Lecture 2 of this series. So what does it mean to be FDA approved? It means spending on average $2.6 billion, 10 years, 1 in 8 chance of success. But at the end of the day, you hope to have 17 years of brand protection where you can charge a lot to recoup those investments. This is the game and how it is played. How does the FDA work? As I said, it's a students pay teachers model, meaning the grades are given out by the FDA, and of course they exercise some independence. But once you get through the system, it behooves them to say, well, it's FDA approved, so if all the untoward side effects or reactions exist, it's just in that really quick fine print that's rapidly spoken at the end of those direct-to-consumer commercials. Yes, people will get hurt, but once you're FDA approved, somehow that should mean something to you, even if you develop one of these untoward effects. I think that's valid. I think it's the best we can do, but realize, of course, just because something is FDA approved, it doesn't mean you won't be one of the people that have a predictable known side effect. So we have to be honest that there is a beltway ecology, meaning in every industry, not just drugs and medical, there is a revolving door between people in corporate business, in academia and research, and in government and lobbying. They all pay each other, they all change jobs together. Not surprising when we look at people in the uh, government regulation of banking, they all come from the banking sector as well. The FDA process involves 4 stages generally. There's preclinical, meaning investigation of new drugs, and then once something seems promising, either with animal studies or small human trials, they might go to a phase 1. This will establish the safety of the compound in question. Phase 2 establishes efficacy, and a phase 3 trial determines different doses. Of course, this model is best suited for a small molecule of a discrete indication, something to block, let's say, the angiotensin receptor or what have you. Even antibodies, yes. But in terms of cell therapeutics, this is not a good model because there is so much variation in terms of cells, and there's such a dynamic system. As I mentioned, the adverse effects are accepted, they're part of the disclosure, and once you make it through the FDA process, you're protected in some regard from litigation. Although, if enough people get hurt by the stuff mentioned in the fast talking or the small print, they'll still shut down a drug if it's causing untoward side effects because in our system there's nothing stopping lawyers from setting up a class action and going after something that is quote unquote FDA approved. Nevertheless, it does serve as a de facto sort of fig leaf for corporations producing drugs that do have some adverse side effects and we hope some disease mitigating benefits. A term known as off label is very important to understand in the context of a patient physician relationship. You've all heard of the lawyer client privilege, and indeed, the greatest power on earth is the power of attorney. Well, I would submit that another great power, once respected but now being eroded by things like legislation, was the patient physician relationship. Now, many substances are used off label since the approval of drugs that go through the FDA was often very narrowly defined. An example of this is the antibody Keytruda, which is mentioned in Lecture 4, that cured Jimmy Carter's cancer. More accurately, Jimmy Carter had metastatic melanoma to his brain, and the Keytruda antibody inhibited the programmed death ligand number 1, allowing his own immune system to overwhelm the melanoma in the way that it was designed to do. Now, the patient-physician relationship is sacrosanct, in my opinion, and even though it hasn't always, in the Marcus Welby days and continuing now into the present, been actually this way, it should ideally be defined by three things. Autonomy, meaning you and I are acting in our own interests. There should be collaboration, meaning we're working together towards a common goal. And most importantly, informed consent, which although it's truly impossible to have a completely informed consent because you and I have different understandings of what everything means and their relative valences, and we have different risk tolerances, the idealized relationship is that the physician is expected to act in the best interests of the patient. That may not always occur, but that is the ideal that we strive for. So, in the sense of the old Covey sort of teachings, Franklin Covey course, it's a win-win or walk away. In other words, patients should never be forced into anything. Likewise, in most cases, physicians should not be forced into anything. It should be a collaborative relationship, and as much as possible, the patient should be informed. Now, turning to the FDA's rulings on cell therapeutics, of course, it's a very difficult subject. The standard that they've come to is that it should be minimally altered. What this means is that you can have a liposuction, and as long as all you're doing is spinning and isolating the adipocyte-derived stem cells, that you can take legally in this country. They figure that very little can go wrong in a single session without storage, collagenase, preservatives, and clonal expansion. So that is currently allowed. Having said that, they're not guaranteeing that it works or that it's worth the money, but they've decided to back off on that for perhaps some greater good reason. Now we look at California's Institute of Regenerative Medicine, which was empowered by a bond issuance. They got billions of dollars, and their mission was, of course, to spend it. And they did, and they spent it on academic and even corporate trials. The benchmarks were somewhat lacking for results, and of course, the question of whether you take a product that seems to be working or a modality, who can own that? Who can make money off of that? Those are questions that really were not addressed. So again, the mission, as in many cases, is just to serve its own existence, not necessarily to provide answers or cures. What this means in practical terms is that when you go to Panama and lay down your money, they can do things that are outside of the FDA purview of minimally altered. We can use collagenase to more efficiently extract the desired stem cells from the matrix of liposuction, and they could even store and perhaps clonally expand those cells, give you billions instead of just thousands back. Uh, and perhaps that is a good reason for the FDA to prohibit that in this country. There are many different points of failure in those processes, which we cannot even go into. So, as I mentioned, autologous fat-derived, adipocyte-derived stem cells is the only therapy available in the United States currently. Although you can, at this time, extract your stromal vascular fraction, take that preserved stem cells, freeze it, and at some later date, thaw them and get them clonally expanded to billions for future use overseas. Uh, Again, I'm not recommending that, but that is a modality that is available if you're willing to take a few steps and travel outside the country. Now, again, looking at CERM as a model, everyone's in favor of research. It keeps food on the table, it keeps the appearance of progress intact, but at the end of the day, all these therapies are still off label until they go through the FDA approval process. And is the FDA even mandated to approve cell therapeutics? They certainly are not sure, and neither are the rest of us. What we do know is that there is a patient-physician relationship that provides some kind of framework for taking these, although many people are quite concerned about how much of a Wild West, an unregulated, and perhaps even at times unsafe environment it can be when introduced with bad judgment, profit motives, and poor clinical methods. Now, why are people even thinking about regenerative medicine? That's because the science of how we understand stem cells to work in all human and animal systems is promising, and indeed some of the clinical trials and anecdotes are also quite astonishing. But the problem is that each person, if you want to be technical, can be a new drug application. In fact, from the same single bone marrow or liposuction, we have many different versions of stem cells. So what are we really talking about in terms of intellectual property? We can't go through an FDA trial for one person's stem cells taken on one day, single use, costing millions of dollars. It makes no sense. So the bigger issues of intellectual property are just the start. There's also issues of quality control, protection of consumers and patients, and safety. Some clinics are injecting them in inappropriate places for inappropriate indications. And indeed, when you thaw them or freeze and thaw and reuse multiple times, you can introduce the risk of infection as well. These are all quite legitimate concerns. There have been bad actors who, again, multi-dose. That's a no-no. When you introduce air, you introduce bacteria. They're single use only. And some people inject them, for example, into the retina, causing blindness. This was probably not a good idea. Poor judgment, poor outcome, and a damage to the whole field of regenerative medicine, in my opinion. Now, turning to the corporate partners and the quality assurance question, one of the founders of regenerative medicine, as far as mesenchymal stem cells, or medicinal stem cells as he calls them, is Dr. Arnold Kaplan, PhD, who founded Osiris Therapeutics. They currently are doing trials on mesenchymal stem cells for disease indications. Also, there's Cytori doing a trial for tissue regeneration. Also, many hundreds of these trials. The future, as in terms of their use in the clinic, their intellectual property protection, their viability as business entities, is quite uncertain. Why people are doing this is because the hope for an MSC product that is FDA approved is very high, based on a lot of real experimental, clinical, and animal data. Perhaps the early frontier products will be for orphan indications or indications for diseases without any good or adequate mitigating cure or drug. There may be for compassionate use, such as the right to try legislation. But hopefully, eventually, we'll get an MSC product that is addressing chronic degenerative changes, which is really what we're all experiencing as we age. Of course, the problems of quality assurance will always exist, as well as the problems with cell engraftment and graft versus host or host versus graft immunogenicity. If you want to learn more about that, see Lecture 6, Are All Exosomes Created Equal? And also Lecture 3 about parabiosis. The way forward perhaps is in the use of exosomes, which give you the benefit of the MSCs without the risk of major histocompatibility reactions. In fact, they are acellular. They do not propagate themselves like viruses. They only are the messages or the songs played for the cells. This means that the therapeutic index is quite high. They will not self-replicate per se, and you could probably take quite a bit without causing harm in most cases, as the MSC actions tend to cluster around decreased inflammation and increased regeneration. Now, the MSCs, as Dr. Kaplan's videos attest, are anti-inflammatory, they're antibacterial, also raising the safety or the therapeutic index. They are anti-apoptotic, and they decrease autoimmunity, again making these a quite safe modality, theoretically and by all accounts, empirically. If the intellectual property is given to process trade secrets, in other words, how they're produced, then certain iterations of exosomes within parameters or specifications could be patented, I suppose. Just like they have the Henrietta Lacks HeLa cervical cancer cell as a lab standard for testing, we could have a single indication FDA trial using exosomes of similar quality and consistency occurring, although, again, perhaps financially, practically, and in terms of clinical application, this might be a non-starter. What isn't a non-starter is that the future of exosomes is big pharma. This article from last year discusses the fact that companies are hoping to use these vesicles to package their little proteins, their RNA. This is a whole other sort of category, but of course, why not mimic what nature does to get drugs around the body? Now, of course, I answered the question, who would want this, in a cynical way, but it's true. If MSC exosome therapy was very helpful for mitigating and reversing disease states, of course, there are certain players that would want it shut down and going through a much more stringent process, perhaps even blocking their availability. I believe research in terms of drug delivery will always be robust because drug delivery is a money-making scheme. Although the natural songs of healing that can be played using exosomes naturally, humanely, ethically derived may not be a business model for everyone. Even though the fact that anecdotes, scientific evidence, and animal research appear to show a wide variety of benefits, we may be waiting for a very, very long time for the FDA to approve exosome uses. So is there a future for therapeutic exosomes? Well, yes, there is. Of course, the FTC will prevent advertising of exosomes to protect consumers. The FDA serves to prevent people from making disease prevention and treatment claims. So this will really be powered by anecdotes, word of mouth, and only used under a fully informed patient-physician relationship. Of course, this is predicated on the fact that it has to be safe, effective, show benefits, and be reasonable and not exorbitant in its pricing. Quality assurance is a problem for manufacturers, so we are dependent upon them doing their job compulsively and ethically. And I think as a general guideline, reuse should be forbidden. Just like you don't thaw pork and refreeze it thawed, you do not do the same with exosomes. So it's critical to keep them in cold chain. They can last up to 6 months at -20 or colder. It's said that they can last 3 days refrigerated, but I would not take that risk. They are not packaged in a preservative like DMSO. It's just in saline. So again, there's no risk from the carrier or preservative or solute. This brings us to the eighth and penultimate lecture, which is what are the risks of exosomes? So thank you for your attention. I'll see you in the next lecture.